Good day to everyone joining us and welcome to today's Xtalks webinar. Today's talk is titled Biomarkers in NASH Drug Development, Using Cytokeratin 18 as a Secondary Endpoint in Clinical Trials. My name is Nima and I'll be your Xtalks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for online streaming. You'll be receiving a link to the recording of this webinar via email in a few business days following this event, and you can also access all recordings you've registered for under your My Profile section of xtalks.com. At this point, I'd like to thank Diapharma, who helped to develop the content for this presentation. Diapharma is a US-based distributor of specialty diagnostic and research assays for the fields of hemostasis and thrombosis, hepatology, pharmaceutical biosafety, and environmental toxicology. With a team of dedicated technical and scientific product managers, Diapharma is able to bring leading assays from the EU to the US market. Their portfolio of biomarker assays include the M30 Apoptosense and M65 ELISA kits from VLV Bio, for research use only in the US and Canada. Now I'd like to introduce our speakers for today's event. Dr. Naeem Al-Khoury is an Associate Professor of Medicine and Pediatrics at the University of Texas Health in San Antonio. He is the Director of the Metabolic Health Center at the Texas Liver Institute. He was most recently Assistant Professor of Medicine at the Cleveland Clinic Digestive Disease and Surgery Institute and Director of the Metabolic Liver Disease Clinic. He completed his medical degree at the University of Damascus in Syria before completing his internal medicine and pediatric residency at Albert Einstein Medical Center in Philadelphia. Dr. Al-Khoury completed his pediatric gastroenterology, hepatology, and nutrition fellowship at Cleveland Clinic and an adult transplant hepatology fellowship at the Digestive Disease and Surgery Institute at Cleveland Clinic. Dr. Al-Khoury received the American College of Gastroenterology Junior Faculty Development Award to study the analysis of breath volatile organic compounds to diagnose non-alcoholic fatty liver disease. Dr. Al-Khoury has over 100 publications and presents his work at both national and international medical conferences. Dr. Al-Khoury is a member of several professional organizations, including the American Association for the Study of Liver Disease, the European Association for the Study of the Liver, and the American College of Gastroenterology. His specialty interests include non-alcoholic fatty liver disease, viral and liver transplantation. Dr. Sabina Payalunga is the Associate Director of Scientific Affairs at Cellular, where she provides scientific support for early clinical studies, client consultation, and subject matter expertise for metabolic disease drug development programs. Her areas of expertise include the obesity, type 2 diabetes, and non-alcoholic fatty liver disease indications. Sabina joined Cellular in April 2015 and previously held the position of metabolic and pharmacodynamic specialist. Sabina received her PhD from McGill University in Canada in the Department of Biochemistry. Her training also includes 3 postdoctoral fellowships at the University of Waterloo and University of Guelph in Canada and Maastricht University in the Netherlands. Over her research career, Sabina has contributed to more than 30 peer-reviewed articles and continues to publish on topics of metabolic disease, pharmacodynamic tools, and regulatory aspects of drug development. Sumit Kaur is the lead scientist for biomarkers and bioanalytical services at Cellular. In this role, he is responsible for the development of immunoassays for biomarkers and biosimilars. Sumit received his BS from Creighton University and has over 10 years of experience in molecular biology and disease pathophysiology. He has authored 5 peer-reviewed publications and numerous posters and presentations on the topics of bioanalysis, biomarkers, and biosimilars. He joined Cellular in 2017 and has an intimate understanding of the regulatory requirements for biomarkers in drug development. So now, without further ado, I'd like to hand the presentation over to our first speaker. Dr. Naeem Al-Khoury, you may start the presentation whenever you are ready.
Hi, this is Naeem Al-Khoury, Associate Professor of Medicine at the University of Texas Health in San Antonio, and my task for today is to discuss the disease spectrum of non-alcoholic fatty liver disease and its healthcare burden. Non-alcoholic fatty liver disease is considered the hepatic manifestation of the obesity epidemic and type 2 diabetes epidemic, and it's considered by many experts a component of the metabolic syndrome, which also includes insulin resistance, dyslipidemia in the form of high triglycerides and low HDL, and hypertension. And NAFLD can contribute to the development of metabolic syndrome, and metabolic syndrome is very common in patients with NAFLD. NAFLD is very common in the United States, so we estimate that in adults it affects about 25 to 30% of the general adult population. If you're an obese adult, then the rate is about 50 to 70%, and then if you look at the bariatric surgery population, severely obese, it's about 80 to 85%. And importantly, in patients with type 2 diabetes, it affects up to 75% of patients with type 2 diabetes. What you see in the figure is a representation of the correlation between the prevalence of NAFLD and obesity in different countries, and you see that there's a linear correlation. Unfortunately, in the United States, we have high prevalence rates of both conditions, NAFLD and obesity. And NAFLD can also affect young adults and children. And what you see in this slide is a study that we published a couple of years back looking at a national database called the National Health and Nutrition Examination Survey, or NHANES database, where we included subjects aged 18 to 35 years. And we looked at them in 3 study periods, starting in 1988 all the way to 2010. We define NAFLD as the presence of overweight or obesity and elevated ALT in the absence of other chronic liver diseases such as hepatitis C and alcoholic liver disease. And by using a low threshold for ALT, so above 30 for men and above 19 for women, we showed that the prevalence of NAFLD in young adults increased from about 10% back in 1988 all the way to about 25% in 2010, and this is in young adults. Other studies have shown that NAFLD also affect about 10% of the pediatric population. It's very important to understand that NAFLD is a disease spectrum that starts with non-alcoholic fatty liver, where you have accumulation of lipids within the hepatocytes, and this is what you see to the left. But there is typically only lipid accumulation, but no inflammation and no hepatocyte injury and no liver fibrosis. NAFLD is considered relatively benign, and typically it does not progress to more advanced disease, and if it does, it takes longer time. Whereas some people with NAFLD have what we call NASH, non-alcoholic steatohepatitis, where in addition to having lipid droplets, you see neutrophilic infiltration of the liver, so inflammatory infiltrate. You see also signs of injury to the hepatocytes in the form of ballooning and variable degrees of fibrosis, which leads to the next step, which we call fibrotic NASH, where you see collagen deposition and you start seeing significant fibrosis. These patients are at significant risk of developing NASH cirrhosis, where you see nodular liver, and these are the patients that may end up needing liver transplantation, and they have high risk of decompensated liver disease. In terms of understanding the natural history of NAFLD, as I said, NAFLD is considered benign, but not completely benign. So we estimate from one systematic review that it takes about 14 years on average to progress by one fibrosis stage if you have NAFLD on the index biopsy. And this is in comparison to patients with NASH, so non-alcoholic steatohepatitis, where the average for progression by one fibrosis stage is about 7 years. So progression happens faster if you have NASH compared to NAFLD. Another aspect of the spectrum of NAFLD is liver cancer, hepatocellular carcinoma. This is a study from the Cleveland Clinic that looked at patients with NASH cirrhosis and patients with hepatitis C cirrhosis. Now, we know that most people that develop liver cancer, typically they have underlying liver cirrhosis. And when they compared NASH cirrhosis to hep C cirrhosis, they showed that patients with NASH cirrhosis developed HCC at an annual rate of 2.6%, which was lower than hepatitis C cirrhosis, but it's still significant, and this meets the threshold for screening for HCC in patients with NASH cirrhosis. More recently, we started seeing cases of HCC developing in patients with NASH in the absence of cirrhosis. And this is a study published by Dr. Al-Sarraj in 2015 looking at the VA system, where they included 1,500 veterans that developed HCC. And then they looked at the underlying liver disease and the percentage of patients that had cirrhosis at baseline. And what they showed that for the most common cause of HCC in the United States, hepatitis C, hepatitis B, more than 90% of patients had underlying cirrhosis. But when they looked at patients with NASH, they found that about 1/3 of these patients developed HCC in the absence of cirrhosis. And this is very concerning for us because we have a large percentage of the population affected with NAFLD without cirrhosis, and we are seeing a rise in liver cancer cases in the United States. So we estimate that if you have, again, advanced fibrosis cirrhosis, the risk of developing cancer is about 2 to 3% per year over a period of 3 to 7 years. It's about 2 to 13%. If you have NASH, then the estimated risk is about 0.1 to 0.2% over a 5 to 6 year period. So definitely lower, but then we have more patients affected with NASH, and this is why we are concerned about seeing more cases of liver cancer. And finally, NASH is becoming a common reason for liver transplantation. And in a recent publication in the American Journal of Gastroenterology, we've shown that NASH is now the most common indication for liver transplantation in women in the United States. So what you see in these graphs are the different indications for liver transplantations in the US and the number of new transplants. And the orange line represents hepatitis C, which historically speaking has been the most common indication for liver transplantation in the United States. But now with the development of highly effective direct-acting antivirals, you can see clearly that since 2013-2014, there has been a decline in the number of transplants done for hepatitis C. And there is rise in NASH represented in the red line, so it is the most common cause in women, and then alcoholic liver disease represented in the blue line, which is the most common cause in male patients, followed by NASH now. So I hope I showed you that NAFLD is common and it could be a serious illness, and that patients with NASH are the ones at higher risk of progression to clinically relevant liver disease. So to diagnose NASH on a liver biopsy, you need 3 components. Number 1 is steatosis, which is fatty infiltration, and you need to have more than 5%. You need to have lobular inflammation, so this is the inflammatory infiltrate typically in the form of neutrophils, and then hepatocellular ballooning, so these are the swollen hepatocytes with relatively pale cytoplasm that you see. This is different from hepatic steatosis. So these are the components of NASH on a biopsy, and we have a score called the NAFLD activity score that grades steatosis from 0 to 3 depending on the percentage of hepatocytes with steatosis, and then we look at lobular inflammation and we grade it from 0 to 3 depending on the number of inflammatory foci, and then ballooning we grade from 0 to 2 based on the number of ballooned hepatocytes. So few ballooned hepatocytes is considered grade 1 and many is considered grade 2. This was initially developed to look at response to therapy in clinical trials. Now today when we get patients referred to us for NAFLD, typically they come because of elevated ALT or fatty infiltration noted on an ultrasound done for other reasons. However, both ALT and ultrasound have limitations. So when it comes to ultrasound, what we're showing here in panel A is a normal patient, and you can see L indicates liver, K is for kidney. So the echogenicity or the brightness of the liver is typically similar to the kidney in a healthy subject, and you can see also the vessels clearly within the liver. When you have fatty infiltration of the liver becomes more bright or echogenic, so this is what we call increased echogenicity, and the vessels within the liver become blurry. So you can see clearly that this is operator dependent and could be subjective. And the sensitivity of ultrasound does not become sufficient until you have about 20% fatty infiltration of the liver, but as we discussed discussed earlier, we consider pathologic accumulation anything more than 5%. So less severe steatosis could be missed on ultrasound. ALT can be also normal in patients with NAFLD. In patients with type 2 diabetes, one study showed that up to 50% of patients with type 2 diabetes and normal ALT can have fatty infiltration of the liver on MRI. And most importantly is that both ALT and ultrasound cannot diagnose NASH or stage the severity of fibrosis. So you cannot determine the severity of fatty liver disease based on ALT or ultrasound. Now, in terms of diagnosing fibrosis, we do have a reliable fibrosis scores, biomarkers, and imaging technologies to determine the stage of fibrosis. And I divide these into serologic markers and imaging tests. And then the serologic markers can be further divided into simple fibrosis scores that rely on readily available clinical and lab characteristics such as AST to ALT ratio, AST to platelet ratio index, and then the FIB4 and NAFLD fibrosis scores. And I'll show you the components of these two in the next slide. And then we have complex serologic markers. These rely on biomarkers of extracellular matrix turnover, things like hyaluronic acid, and these include tests like FibroSure, FibroTest, or the ELF test. And then the imaging test, they measure actually liver stiffness with the concept of elastography, and this includes vibration-controlled transient elastography, MR elastography, and other technologies. So here I'm showing the NAFLD fibrosis score, which is an online calculator. All you need is to know the patient age, BMI, presence of diabetes or insulin resistance, liver enzymes, platelet count, and albumin. And then you get a score. You can have a low score indicating the absence of significant fibrosis or a high score predicting the presence of significant fibrosis. But then some values fall in between, and this is what we call an indeterminate score. The fibrosis 4 or FIB4 score relies on age, AST, ALT, and platelet count, and you can divide the patients similar to the NAFLD fibrosis score into low risk, high risk, and then indeterminate. So these are easy scores that we can calculate in our clinics. In terms of imaging technologies, again, we have transient elastography with FibroScan. We have other ultrasound-based technologies such as ARFI or acoustic radiation force impulse, and then MR elastography. They all have advantages and disadvantages, but they're reliable to rule out the presence of cirrhosis and advanced fibrosis. So as you can see, we can diagnose fibrosis in a reliable way, but we do not have a good non-invasive way to diagnose NASH, and it's really not feasible to biopsy every patient with non-alcoholic fatty liver disease to determine if they have NASH. So what you are seeing here in the CAT scan image is a big hematoma after a liver biopsy. And again, because this is a very prevalent disease, we cannot biopsy all these patients. So there's an urgent need to develop non-invasive biomarkers for the presence of NASH. And to develop biomarkers, you need to understand the mechanistic pathways that lead to disease progression from steatosis to NASH and fibrosis. And in fatty liver disease, the first step is the accumulation of fat within the hepatocytes in the form of triglycerides. This is what we call steatosis. But once you have these steatotic hepatocytes, they become more susceptible to secondary injuries. They have increased endoplasmic reticulum stress, mitochondrial dysfunction. All these factors predispose the hepatocytes to apoptosis, which is programmed cell death. And when you have these apoptotic hepatocytes, this can induce the hepatic stellate cells, and these are the fibrogenic cells that produce collagen and lead to fibrosis, and can also activate Kupffer cells, the resident macrophages in the liver. So hepatocyte apoptosis plays a significant role in transition from fatty liver to NASH. And in a landmark study published by Feldstein et al. In Gastroenterology back in 2003, they showed clearly that patients with NASH, steatohepatitis, they have increase in hepatocyte apoptosis, and this is what you see highlighted by the arrow. This is an apoptotic hepatocyte. So patients with NASH had more hepatocyte apoptosis compared to normal controls, but also patients with NAFLD or just simple steatosis. And luckily, we can actually determine an increase in hepatocyte apoptosis in the peripheral blood by using biomarkers such as cytokeratin 18, which will be the topic for further discussion. So when the hepatocyte undergoes apoptosis, there's activation of caspases, and caspase 3 is what we call an effector caspase that can cleave certain intracellular components, including cytokeratins. So cytokeratins are the major intermediate filaments within the hepatocytes, and when the caspase 3 cleaves cytokeratin 18, you have the release of these cytokeratin 18 fragments into the blood, and then we can actually measure them using a simple ELISA with an antibody, so we call it the M30 ELISA. And in a landmark paper published in 2006, they evaluated the utility of plasma CK18 levels to determine the presence of NASH in patients with NAFLD, and they showed clearly that patients with NASH had higher levels of cytokeratin 18 fragments in the blood compared to patients with NAFLD or steatosis and patients with normal liver biopsy. So this is just a brief introduction to cytokeratin 18 and the disease burden of NAFLD NASH, and now I'd like to hand it over to Sabrina to provide a more in-depth discussion of cytokeratin utility in diagnosing NASH. Thank you.
Thank you so much, Dr. Alkhouri, for that fantastic summary of NASH and its non-invasive biomarkers. Hello, everyone. I'm Sabina Payalunga, Associate Director of Scientific Affairs at Cellular. Cellular is an early-phase CRO active in the NASH space, and we offer a number of non-invasive measures of NASH assessments like FibroScan and soluble biomarkers for early clinical studies. In this next section, I'll focus on CK18 as a biomarker in clinical trials. But before I begin, I just want to provide a brief background on cytokeratins, also known as keratins. They are proteins that assemble into filaments, and they provide integrity to cytoskeleton and support the cell structure. They are abundant in epithelial cells and undergo a number of post-translational modifications such as ubiquitination and phosphorylation. They actually act as a phosphorylation sink or a decoy binding site for phosphorylation-dependent stress responses. Cytokeratins are also major caspase substrates. So because of this, they're thought to have a role in cell protection. By taking the brunt of cell stress, they allow the cell to survive and adapt. But because they are targets of cell stress, they are also associated with a number of diseases. And here is a picture showing just some of the diseases associated with various cytokeratins. In the liver, CK8 and CK18 are major components of Mallory-Dank bodies. This is a histological feature that is observed. It's an inclusion body that has a number of aggregates of misfolded and ubiquitinated proteins, which is typically observed in biopsies from NASH patients. CK18 also serves as a soluble biomarker of cell death. And in a necrotic cell, we have loss of cellular integrity. So the CK18 can leak out of the cell. Here, an antibody directed against the full-length CK18 can be used as a biomarker of necrosis. Apoptosis, on the other hand, is an organized or programmed cell death. It's caspase-mediated, and the caspase-cleaved form of CK18 can be detected with an antibody called the M30 antibody. And our next speaker, Sumit, will talk a little bit more about these antibodies. The caspase-cleaved CK18 is increased with the progression of fatty liver to NASH, representing the inflammation and apoptosis underlying the steatohepatitis. And here you can see a stepwise increase in serum caspase-cleaved CK18 from healthy individuals to those with fatty liver and NASH patients. And here the CK18 fragment refers to the caspase cleaved CK18. Now, a number of different studies have evaluated CK18 as a diagnostic biomarker in NASH. And a recent meta-analysis summarized caspase-cleaved CK18 cutoff values as well as diagnostic value from cross-sectional studies with biopsy-proven NASH patients. Overall, caspase-cleaved CK18 had good performance in predicting NASH when compared to liver biopsy results, with an area under the receiver operating curve of 82%. And just to compare to other soluble biomarkers, for example, the PRO-C3 also has an area under the curve of around 80% as well. Now, moving on to clinical trials for drug development, you may be surprised to learn that there are over 80 drugs in clinical development right now. Yet there are no FDA-approved drugs for NASH. Many of these drug programs have a fast track or accelerated track designation, meaning that they'll rely on a surrogate endpoint. For phase 2B studies, previously accepted surrogate endpoints include a 2-point or more improvement in the NAS score. And just to remind you again, NAS is the NAFLD activity score, which has the 3 components of steatosis, ballooning injury, and inflammation. For phase 3 or approval, surrogate endpoints include the resolution of steatohepatitis with no worsening of fibrosis, improvement of fibrosis with no worsening of steatohepatitis, or both. And here, resolution of steatohepatitis refers to 0 for ballooning or 0 and 1 for inflammation. Now, for early clinical phase studies, the FDA published a set of recommendations in association with the American Association for the Study of Liver Disease in 2015 and recommended primary endpoints of safety and tolerability, as well as reduction in hepatic steatosis by magnetic resonance. Key secondary endpoints include decrease in ALT as well as changes in triglyceride levels, and then a reduction in markers of cell death and injury, such as CK18. More recently, the Liver Forum recommended a set of baseline biomarkers to be evaluated in clinical trials for NASH. The Liver Forum is a consortium of industry, academic, and regulators aiming to advance the regulatory science of NASH. And they listed a set of biomarkers to be able to track and understand better the natural history of disease as well as their effect upon treatment response. And these biomarkers are for oxidative stress, fibrosis, inflammation, as well as CK18 as a biomarker of apoptosis. The Liver Forum also compared CK18 to other non-invasive NASH biomarkers, such as clinical chemistry panels or combined biomarkers, as well as ultrasound and the FibroScan CAP, and found the criteria to be quite similar among all these modalities. The magnetic resonance imaging or magnetic resonance spectroscopy does remain the gold standard for detecting steatosis as a non-invasive modality. And here's a snapshot of how CK18 is being used in clinical trials. You can see the majority of use is within phase 2. And that makes sense because this is the first time that a drug is typically evaluated in a patient population. There's only a handful of drugs that are currently in phase 3 development. So as more drugs enter into phase 3, it's anticipated that this slice of the pie will probably increase as well. And I mentioned that the NASH pipeline is quite robust. And here's an example of some of the investigational drugs and their targets for NASH. And overall, they can be kind of divided into three main types or three main functions. Those that have an effect on metabolic pathway, all shown here in green. Those that affect the anti-inflammatory pathway, shown in black, and then anti-fibrotic agents, which are highlighted in blue. There's three drugs in particular that I wanted to focus in on, and that's selonsertib and emericassin, which are anti-apoptotic agents, as well as cenicriviroc, which is an anti-fibrotic drug. Selonsertib is an apoptosis inhibitor, and in a phase 2 study, efficacy of selonsertib alone or in combination with simtuzumab was evaluated in an open-label 24-week study. Simtuzumab is a putative anti-fibrotic agent, and that in previous studies showed no clinical benefit. So for the sake of this analysis, it was treated as a placebo, and the selonsertib groups were pooled. The results showed that there was a dose-dependent effect in the number of patients with improved fibrosis. So 43% of patients showed improved fibrosis on 18 milligrams of selonsertib, while 30% in the 6 milligram group, and then 20% in the control. While the percent patients with improvement in the NAS score was similar in all three cohorts, the 18 milligram group had greater proportion of patients with inflammation, and this tracked with greater reductions in both the M65 and M30 CK18 measurements. In a sub-analysis, the change in CK18 according to fibrosis response was evaluated. And here we have in green those that had improvements in fibrosis, and in red those with no change or worsening of fibrosis. And you can see that there's a greater CK18 reduction with fibrosis improvement. Similarly, there was a greater CK18 reduction with inflammation improvement. And next, in a proof of mechanism study, the pan-caspase inhibitor emericassin was evaluated in a placebo-controlled study with 38 subjects that had NAFLD and elevated ALT. Emericassin had greater ALT reductions after 7 and 28 days. And as a way of looking at target engagement, 28 days of emericassin treatment also resulted in a reduction of about 30% of the M30 and M65 CK18 measurements. Caspase activity was also reduced, therefore showing target engagement. And in a recent follow-up study in a small group of 11 NASH cirrhosis patients, they found that CK18 was reduced, the caspase cleaved form of CK18 was reduced after 1 and 2 months of treatment, and the full-length CK18 was reduced up to 3 months. The final study I wanted to review is the CENTAUR study, and this is a phase 2b study with cenicriviroc, which is a chemokine receptor inhibitor. This study examined cenicriviroc over 1 and 2 years of treatment, as well as the NASH progression in placebo over 2 years in NASH biopsy-proven patients. And the one-year results are published, so we'll discuss those. After 1 year, the primary endpoint was not met. There was no change in NAS and no worsening of fibrosis for Cine-Crivi-Rock and placebo. But the key secondary endpoint was met, and there was a greater proportion of subjects that had improvement in fibrosis by 1 stage and no worsening of steatohepatitis with the Cine-Crivi-Rock group compared to placebo. Drilling down into the NAS components, for inflammation, you could see that the percent of patients with improved, no change, or worsening for inflammation were similar between Cine-Crivi-Rock and the placebo groups. And for ballooning injury, there may be a slight favoring of improved ballooning in the placebo group. And this slight favoring with the placebo group seemed to translate into a greater CK18 reduction in the placebo group than the Cine-Crivi-Rock. And it's important to know that the NASH has a natural progression that waxes and wanes, which can affect the placebo response. Also, there's factors such as genetics, diet, exercise, even coffee consumption, that we don't know exactly how that impacts the placebo response. And more research is needed on this front, as well as strong controls for the placebo group. Now, cellular injury has also been assessed in drugs other than those targeting just anti-apoptotic pathways. And here you can see that there has been positive treatment effects seen with obeticholic acid, which is a synthetic bile acid, as well as leptin therapy. In addition, Piven's study, which looked at pioglitazone and vitamin E treatment, also found reductions in caspase-cleaved CK18 at 16, 48, and 96 weeks of treatment. And this reduction in CK18 was associated with improvements in inflammation and ballooning injury by histology. So in summary, we've seen that there's a strong association between CK18 and NASH, as it's a biomarker of apoptosis and a key marker of steatohepatitis. CK18 is a valuable secondary endpoint for drug development and recommended as a biomarker of a cell injury, as well as proof of mechanism for anti-apoptotic drugs, and changes can be observed with other drug mechanisms as well. However, it's important to note that for use as a secondary endpoint, assay validation is required. And my colleague Sumit is now going to address how biomarker can be validated. And so I'd like to pass the baton on to Sumit.
Thank you, Sabina. So today I'll be speaking about the validation of CK18, the apoptosis biomarker, the assay for NASH drug development. I'm Sumit Kaur, lead scientist for biomarkers at Bioanalytical Services at Cellular. So as Sabina mentioned, keratins are a commonly used biomarker in many tissue types, and they're expressed in many epithelial cells, but hepatocytes are unique in that they express only K8 and K18. This makes it a slightly more specific marker for hepatocyte cell death. So looking more specifically at hepatocyte cell death, alcohol causes necrosis, which is unregulated and inflammatory cell death. It causes the leakage of full-length keratin 18 fragments into the serum, and this is detected by the M65 ELISA. NASH, on the other hand, leads to apoptosis, which is a regulated cell death, and leads to a caspase-cleaved release of CK18 fragments. These fragments are specifically detected with the M30 ELISA, which is what we used in our validation. So as Sabina has mentioned, CK18 is an effective biomarker in NASH trials, and it's used most commonly as a secondary endpoint in these trials. However, before an assay can be used for drug development and clinical data, the assay itself must be validated, which is what I'll be describing. So I wanted to speak a little bit about the guidelines dictating how an assay should be validated, specifically biomarkers. So for PK assays, there are many detailed guidelines from regulatory agencies such as the FDA and EMA. For clinical biomarkers, there are similarly detailed guidelines from CLIA, CAP, and CLSI. However, for biomarkers in drug development, the guidelines are less clear. The 2018 BMV guidance from the FDA has a small section on biomarker assay validation, and there are several other white papers on this topic. So the FDA BMV guidance says that when a biomarker is used for supporting a regulatory action for a drug, the assay should be fully validated in a bioanalytical lab. This validation should be fit for purpose based on the context of use, and that context of use should be determined by the sponsor. And I'll be describing context of use in a few slides. It also describes recommendations for adapting a commercial kit when using it for drug development. And we followed these guidelines because we did adapt the M30 commercial kit. Finally, it suggests that biomarker data, when used in a clinical environment and not a bioanalytical environment, may be rejected. So why should a bioanalytical validation be performed? Well, there's a scientific justification and a compliance justification. So for clinical chemistry assay, these assays are different from a bioanalytical assay. A clinical chemistry assay is designed to distinguish from normal and abnormal values. And as you get farther away from the cutoff of the assay, the variability in the assay increases significantly. And that variability can be greater than 20% as you get farther from the cutoff. A bioanalytical assay, on the other hand, is designed with constant variability. The standards and QCs are typically prepared in the study matrix, the matrix of the study of the trial. And the standards and QCs are designed so that the variability is constant within 20% over a large range. And that large range is needed for a drug development trial when you are looking for changes in concentration. There is also a compliance justification for using bioanalytical assays. Data retention, for example, is dictated by the 21 CFR Part 11 compliance. And batches in a bioanalytical assay are also different. Bioanalytical assays use quality controls and QCs on each batch. And the batches can be designed so that they're from a single study or a single patient. So, as I've mentioned, biomarker validation for an assay must be fit for purpose based on context of use of the biomarker. So what is context of use? Well, context of use defines how the biomarker is used in the trial specifically. So it can be used from discovery all the way from drug launch. So in discovery, a biomarker is typically used to distinguish normal versus abnormal. In a trial, the goal is to determine a treatment response. And in later stages, a biomarker can also be used for dosing and pharmacokinetics, or PK. This can also be looked graphically, looking at the importance of the biomarker and the tests needed to validate that biomarker. This can be grouped into 3 tiers, tier 1, 2, and 3. So in earlier tiers, tier 1, less validation is needed because the biomarker is only distinguishing normal from abnormal. Versus in a tier 3 supporting a regulatory claim, more validation tests are needed as the biomarker is more crucial to the study. So this is a busy slide, but it details the tests that are needed, or at least the recommendations of tests that are needed for each of the 3 tiers of validation. As you can see, in a tier 1 exploratory validation, less tests are needed to validate the assay. In a tier 3 full validation to support a regulatory claim, more tests are needed, such as using an international standard reference material, if available, performing parallelism, and performing more precision and accuracy batches. So for CK18, we performed a Tier 2 validation, and this was for a context of use as a secondary trial. As Sabina has shown, that is most commonly what it's used for. So the test that we used for the validation was to make an endogenous QC from study matrix to support the recombinant QC supplied in the kit. We performed 3 precision and accuracy batches, matrix effect testing, parallelism, and stability testing. So the CK18 method was an adaptation of a commercial kit. This was the Diapharma M30 Apoptosis ELISA. It's a research use only kit that we adapted for drug development. Now, adaptation of commercial kits is an entirely large topic, could be a separate talk, but I'll just briefly mention that we adapted it by creating a 6-point standard curve. We made QCs internally from reference material to replace the kit QCs. And we collaborated with the vendor to assist in validation data to support our own validation data. So the first validation test we performed was to do precision and accuracy. We performed 3 batches on separate days, and we looked at the precision and accuracy of QCs and an endogenous QC in study matrix. So as you can see, precision was within the guidelines of being less than 20% variability, as well as accuracy was within 20% for all of the QCs. As part of precision and accuracy testing, we also validated the sensitivity of the assay. We verified it from 64.7 units per liter to 964 units per liter. This fits with vendor data, and recently the M30 kit was actually improved for sensitivity. The background was reduced in the assay, which improved sensitivity. Now, clinically, this is important because some NASH studies have used the older version of the M30 kit or have used other CK18 kits. And these suggested that CK18s may not be a useful biomarker in NASH. However, newer studies which have used the improved version of the kit have shown usefulness of CK18. So matrix effect is the next test we performed, and matrix effect tests for interference from endogenous compounds. And this is especially important in later stage trials because patients have components in their serum and concomitant medications that may interfere with measurement of CK18. So we measured CK18 by spiking different amounts of reference material into endogenous serum and measuring the measured concentration. And our measured concentration was within plus or minus 20% of the expected concentration, which fits within guidelines. The final assay I'll be talking about is parallelism. And parallelism is an important assay for biomarker validation because for biomarkers, the reference material in a kit is usually a recombinant reference material. And this is different and can be different from the endogenous biomarker that is being measured in the body. So parallelism has dilutions of both the reference material and the endogenous biomarker, a high concentration study sample, several dilutions, and if parallelism is achieved, the signal to response of the recombinant material should be similar to the signal to response of the endogenous biomarker, as shown in panel B in this slide. So we performed parallelism of CK18 with 3 different dilutions using a high concentration study sample in human serum. And as you can see in the graph, the measured concentration was consistent for all 3 dilutions, showing that parallelism was achieved and the recombinant CK18 material, reference material, is a suitable reference material for endogenous CK18. Finally, we measured CK18 concentrations in both healthy and NASH samples. These are commercial samples that we purchased. And in the samples, we see that there's a significant increase in CK18 in NASH samples. These are all stages of NASH. They were grouped together. And the significant increase demonstrates that the assay has significant analytical performance to detect changes from healthy and NASH patients. So finally, I'd like to mention, and Sabina has shown a similar slide, that CK18 is a part of the pathogenesis of NASH, which includes oxidative stress, apoptosis, inflammation, and fibrosis. Each of these processes have different biomarkers, and CK18, we believe, will be part of a panel included in this pathogenesis. So in summary, biomarker data for drug development should be validated fit for purpose based on the context of use for the biomarker. And this can improve the standardization of the assay as well as reduce drug development cost. The CK18 method that we validated fits the context of use of a secondary endpoint in clinical NASH trials. And I'd like to thank the people who assisted with our validation. And with that, we have some poll questions.
All right, everyone, before we move forward with our Q&A session, we have 3 poll questions for our audience members. Now, our first poll question has been launched on your screen, and I encourage all audience members to vote in real time. This poll question is, do you currently use CK18 in your NASH clinical trials? And our options are yes, no, and don't know. All right, perfect. It looks like most audience members have voted on this question. Our second poll question will be launched on your screen right now. Now, this poll question is, if you use CK18 in your NASH clinical trial, at what stage is it used? Now, we have 3 options to choose from here. A, phase 1 and 2 trials only. B, all clinical trial phases. Or C, I do not currently use CK18. All right, perfect. It looks like most audience members have voted on this question. And now it's time for our last poll question before we move forward with Q&A. So this question is, if you are using CK18 in your NASH clinical trial, how is it used? A, secondary endpoint. B, pre-screening to reduce screen failures. C, both A and B. Or D, I do not currently use CK18. All right, it looks like most audience members have voted on this question. Now let's move forward with our Q&A session. Okay, so it looks like we already received a few audience questions, so let's start with those. Okay, so our first question for our speakers is, just wondering if CK18 can also be used as a biomarker for primary sclerosis chylomicronemia for PBC?
This is Sabina. I can take that question. And the surrogate or primary endpoint for primary biliary cholangitis or PSC is really a change in the alkaline phosphatase. However, CK18 could be applied as an exploratory biomarker, especially if you have an anti-apoptotic agent that you are investigating.
Yeah, this is Naeem Al-Khoury. I agree with Sabina, and there are a few papers looking at CK18 in patients with primary biliary cholangitis, PBC, as a biomarker for developing liver-related outcomes or even advanced fibrosis, because hepatocyte apoptosis is an important mechanistic pathway that leads to fibrosis progression. And some of the NASH medications that we are using, for example, selonsertib with antifibrotic effects, they also have anti-apoptotic effects. They are being looked at for other indications outside of NASH, such as PSC, primary sclerosing cholangitis.
Thank you so much for those answers. Our next question here for our panelists is, can CK18 predict fibrosis progression? What are the best fibrosis progression predictive biomarkers up to your knowledge?
I can chime in on that question. So CK18 really tracks with the changes in steatohepatitis, and then because of improvements in steatohepatitis, you could have effects on fibrosis progression. So it's not a fibrosis biomarker per se, but it could track with improved changes there. Looking at biomarkers of fibrosis, Pro-C3 is a common one, as well as Dr. Al-Khoury had listed a couple other biomarkers like hyaluronic acid, as well as others.
Yes, so there's also the ELF score, which includes other biomarkers such as tissue inhibitor of metalloproteinases and also C3 as a component, but a different kind than the C3. And then we have imaging studies. However, predicting the stage of fibrosis is different from predicting fibrosis progression. And even with fibrosis, it's not a one-way street. So some people can progress with fibrosis, some people can regress being in the placebo arm of clinical trials. So there are many factors that play into progression and regression of fibrosis.
Thank you so much for those answers. Our next question here is, this one is actually for Sabina, and it says, what is considered a meaningful decrease in CK18?
So, to my knowledge, there's no data out there yet that shows a change of CK18 with hard outcomes like time to cirrhosis or liver transplants or other hard outcomes. So I think we would get that data once we have more longitudinal studies done, or these phase 4 studies with CK18 as a biomarker. But that being said, we do know that selonsertib data showed that with each 100-unit decrease in CK18, there was an association with fibrosis improvement. And that also a 30 to 40% reduction from baseline in CK18 was associated with improved inflammation.
Thank you for that answer, Sabina. We have another question for you, and this question is, what are the gold standards for primary endpoints accepted by the FDA?
So I can take this. This is Naeem Al-Khoury. So, today, I think, what's accepted by the FDA in terms of getting a drug approved will be based on histology and improvement in NASH. And that could be defined as the resolution of NASH on a repeat liver biopsy after the intervention. And that would be the absence of hepatocyte ballooning and maybe the presence of no inflammation or minimal inflammation and no worsening of fibrosis, or the other endpoint will be improvement in liver fibrosis by 1 stage and no worsening of NASH. So these are the two primary outcomes of the trials that are in phase 3 development. And even liver biopsy is still considered a surrogate endpoint. So the plan is to follow these patients for 5-6 years, depending on the study, to look at the development of hard outcomes such as need for liver transplantation, liver-related decompensation, and overall mortality. Now we are working diligently on getting other approved endpoints outside of histological endpoints, such as improvement in MR elastography or proton density fat fraction or PDFF and other biomarkers. However, today it still relies on liver biopsy and histological improvement.
Thank you for that answer. Our next question here is, how quickly would you expect to see a change in CK18 with treatment?
Well, that would depend on the drug mechanism. For example, in the case of emricassin, which is a direct caspase inhibitor, the caspase cleaves CK18 can be reduced within hours of treatment. With selonsertib data, we saw that after 24 weeks there was reductions, but this was a one-time measurement. Earlier studies like PIVENS showed reductions with 16 weeks. So that's kind of all the information we have now. As more data becomes published, we'll have a better idea of how quickly we'll start seeing changes in CK18 and how they track with histological changes of inflammation and ballooning injury.
Thank you so much for that answer. Now we have enough time to squeeze in one more question, and this question's for Sumit. This question is, are any steps being taken to qualify CK18 for NASH through the FDA Biomarker Qualification Program?
Yeah, this is Sumit. So the FDA Biomarker Qualification Program is a program that qualifies both the clinical use and the analytical validity of a biomarker for all clinical trials. So CK18 specifically has not yet been submitted to this FDA biomarker qualification program. The M30 and M65 kits are being considered for FDA clearance for diagnostic use. And there are groups such as Litmus and Nimble that are considering submitting CK18 for the FDA biomarker qualification program, but this has not been done yet.
Well, thank you very much for those answers. We've reached the end of the Q&A portion of this webinar. If your question wasn't attended to during the Q&A section, the team from Dia Pharma will do their best to follow up with you after the webinar. You can also reach out to our speakers by using the email address that is displayed on your screen right now. Thank you, everyone, for participating in today's conference. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen, and your participation is appreciated as it will help us to improve our webinars. I also just sent you a link in your chat box where you can share this webinar with your colleagues on LinkedIn. Now, please join us in thanking our speakers by sending a quick message through the chat panel. We hope you found this conference informative. Have a great day, everyone.